So what we see here is with displacement of the stereocilia on the apical surfaces of the hair cell, there is the cyclical depolarization and hyperpolarization that happens as the stereocilia are moved back and forth or up and down in the direction that is parallel to the axis of orientation from smallest stereocilium to largest stereocilium. And if one were to move these hair cells in the orthogonal direction, that is rather back and forth from the axis of shortest to longest, then what we would find is basically no change in membrane potential, because we would neither be stretching those tip lengths nor would we allow them to relax back. So if we were to look down on the hair cell, what we would find is that there is a very special type of stereocilium called a kinocilium. We're actually born with this, but for most inner hair cells, the kinocilium is lost in life. Nevertheless, there is this gradient from shortest stereocilium to tallest stereocilium that is present next to where the kinocilium once was. And so if we move these stereocilia along this axis from smallest to longest and back and forth, that's where we find the greatest amplitude of oscillation in membrane potential. If we were to move in the orthogonal axis, basically we wouldn't be stressing out those tip lengths, and consequently we would have no change in membrane potential. Now let's look at how good these hair cells actually are at faithfully reproducing the cycles of pressure that cause these traveling waves to be set up along the length of the basilar membrane. If we stimulate these hair cells at a frequency of, let's say, 300 hertz, 300 times a second, we see a beautiful modulation in the membrane potential of that hair cell. And as we advance the frequency, what we find is that these hair cells can faithfully represent the temporal aspects of the stimulus all the way up through at least a kilohertz. So here's 1,000 hertz, a kilohertz of modulation, and we can still detect a beautiful sinusoidal type cycle in the membrane potential of that hair cell. But once we get above about a kilohertz or so, we begin to lose the ability to follow the frequencies of that pressure wave. And that's a pretty amazing accomplishment, that we can record with fidelity the movements of these hair cells up to more than 1,000 cycles per second. But at some point we just can't operate as quickly as the energy in the environment. But we can still encode that energy because there is a DC component to the response. That is, the hair cell on average becomes depolarized, even though the modulations, the AC component, now basically vanishes as we get above about 2 kilohertz. So as we continue to increase in pitch, in the frequency of the stimulus, we can still activate hair cells in an appropriate zone of the basilar membrane, but we can no longer produce an AC component of modulation that faithfully captures the temporal dynamics of the stimulus. Let's back up for just a moment, and I'll remind you then that at the basal region of the hair cell we find synaptic vesicles. So for an action potential to be fired in the afferent nerve fiber, neurotransmission must take place, and a sufficient depolarization to activate voltage-gated sodium and potassium channels must be generated here in the afferent nerve fiber. Now, I'd like to invite you to pause this tutorial and look at your tutorial handout for a couple of web links to some really excellent resources that have been generated that summarize these aspects of auditory physiology that relate to these peripheral structures found in the middle and the inner ear. The first is an animation found on the website that supports our textbook, and you can navigate there on your own, or you can simply click the link in your tutorial notes. The second is really one of my favorite animations in all of neuroscience. It's just a wonderful tour through the biomechanics of the basilar membrane created by one of the real giants in the field of auditory neuroscience, Dr. James Hudspeth. Well, he didn't actually do the animation, but he commissioned it and oversaw the production of this really beautiful presentation that I think really makes very clear how the basilar membrane can decompose complex sound, even box, staccato, and fugue, into component frequencies that are then encoded in the oscillations of membrane potential in the hair cells that happen to be at each location along the length of the basilar membrane. So please go watch these two animations and then come back and we'll conclude this tutorial. Okay, well, I hope you enjoyed that. I know I sure did. Now, I want to just conclude by talking about sensory coding in the auditory nerve. If we look at the cycles of stimulation that are going to be transduced by our hair cells, what we'll find is that for any given axon in the auditory nerve, there is a barrage of action potentials that can be recorded with some predictable temporal relationship to the stimulus. In this case, it's a fairly low frequency stimulus, and what we see is a faithful and highly reproducible barrage of action potentials that is elicited at a particular phase relationship to the stimulus. If one were to look across a family of axons within the auditory nerve and plot the firing rate that is observed for any given frequency of stimulation, what we would be able to do is to plot a tuning function for that axon. And so what we're looking at here is a plot of the intensity of the stimulus that's necessary to elicit action potential firing in the axon. And what we find, for example, this axon that is plotted here in purple above, is that the threshold is lowest at a particular frequency, meaning it takes less amplitude of modulation to elicit action potential firing in that axon. Well, a different axon is going to have a different best frequency, and so on. So here we have examples from 6 different axons recorded in an auditory nerve, and they all have a different best frequency. And this figure is illustrated in a way that is meant to organize these best frequencies across that tonotopic map that we find established in the basilar membrane. And sure enough, the auditory afferents that innervate the base of the cochlea are going to have their best frequency near the higher range of what can be heard and transduce. So in this case, this gold-colored plot is showing the best frequency around 10 kilohertz for this particular axon that's innervating the base of the cochlea. Meanwhile, way at the other end of the cochlea, here is an axon that's innervating some domain of the basilar membrane that's closer to the apex, and we find a much lower best frequency, much less than 1 kilohertz. Well, I should have made this point earlier, but for us humans, we're sensitive to sound over a range from about 20 hertz to about 20 kilohertz. So if we were to be very close to the base of the cochlea, that's roughly where we would find our maximum frequencies that could be represented across our basilar membrane, whereas out near our helicotrema, near the apex of the cochlea, that's where we would find the lowest range of frequencies found. Okay, well, in our next tutorial, I'd like to discuss with you how this information that is generated here in the inner ear is processed in the central nervous system. So I'll see you back for that discussion.